It's a wild world out there. So many plants and animals, bugs and birds, mushrooms and microbes. There is so much biodiversity out there. How do we keep track of it all? And what's the use anyway? The truth is, we depend on these species for so much. The plants clean our air and reduce the carbon dioxide in the atmosphere. The mammals provide us food and spiritual connections. The bugs, mushrooms, and microbes are nature's recyclers that keep our ecosystems humming. All of these are ecosystem services that we depend on every day. Measuring biodiversity helps us monitor the natural world. This information can help us to see where life is thriving, where it's struggling, and hopefully figure out what the problems are. We do this so we can manage our own species' impact on the planet and save other life from extinction. My name is Dr. Roland Kays. I'm a scientist with North Carolina State University and the North Carolina Museum of Natural Sciences in downtown Raleigh. As an ecologist, I study wild animals, learning new things about these amazing creatures and trying to discover how we can help them survive on our rapidly changing planet. I've had the fortune to work with research groups studying mammals all over the world, including my PhD research with kinkajous in Panama, and later work with lions and bats in Africa. I was also part of the team that described a new species of mammal, the olinguito. We used museum specimens and we also got to study them in Ecuador. Now, I run the Biodiversity Lab at the North Carolina Museum of Natural Sciences, where we study how species are adapting or not adapting to human-caused changes in the landscape. For example, locally, we're looking for weasels, which seem to be declining in North Carolina. But I also work on coyotes, which is a non-native species in the state that is now so common it's causing problems by predating on bird and sea turtle nests. My favorite way to study biodiversity is with camera traps. These are motion-sensitive cameras that we can leave in the woods for months at a time to record whatever animals walk by. At one study site, we typically set out a grid of about 30 cameras to sample the local animal population. By recording the animals in each set of pictures, we can see which species live there and how common they are. But cameras don't work for everything. Different kinds of plants and animals require different survey techniques. For birds, well, we do get birds on our camera traps sometimes, but standard visual surveys known as point counts are better. Here, you just stand at one point for a set period of time and record every bird you see or hear. These days, we also have automated acoustic recording, which is another great option for birds, especially now that artificial intelligence can help us identify which birds are singing in the recordings. You can just leave the microphone out to record all the bird calls over a period of time. These are basically the sound version of camera traps. And we can also use these to monitor frogs or insects that regularly make noise, or even bats if you add a special ultrasonic microphone to pick up their echolocation calls. Fish, insects, plants, mushrooms, spiders, there are so many different types of life on Earth, and scientists have created specialized methods for monitoring each of them. The key thing is to have a standard protocol to get a count of exactly what species live at a given location at a given point in time. Once you have your biodiversity measurement, the next step is to use it to make comparisons. If you measure the same place multiple times, then you can make temporal comparisons to see if an animal population is growing or declining. Or you can take the same measurement at lots of different places, and then you can conduct a spatial analysis to see what factors like temperature, habitat type, or human disturbances affect a species. The ability to make these comparisons is what makes this kind of research so interesting and important. Now, for a grid of camera traps, we can record the number of species detected. We can say in what percentage of cameras each was found, and this is something we call occupancy. Or we can just count up the total number of pictures of each species as a measure of their relative abundance. For example, we wanted to see what the effects of human development were on wildlife. So we set out cameras along a gradient from urban to suburban to rural to wild. And in this case, we were working in Montana, so we could extend our camera tracks from downtown Missoula to some of the most wild wilderness in the United States. Our cameras did find a lot of mammals living in the city, especially white-tailed deer, which were super common. But overall, we found a steady decline in the diversity of mammals as human density increased. This was especially the case for the larger species like moose or wolves and mountain lions, which were only found in the least disturbed sites. Our study showed that while some species can live next to us in cities, it's also really important to protect and preserve the truly wild places to save the full range of biodiversity. Closer to home, we've been working with NC State students to monitor animals locally at Schenck Forest. Each semester for the last 10 years, students have set a grid of camera traps out in the forest. They then go through the pictures to identify the animals, giving us standard counts. In this case, the size of the forest hasn't changed, but we did notice a big change in the coyotes. From 2013 to 2020, we had just a couple pictures of coyotes at a few cameras. But then there was a spike in coyote pictures for the last 4 years. What effect did they have? Well, coyotes hunt deer, especially fawns, so maybe the deer population has declined? No, actually there are about the same or even slightly more deer recently. But it's a different story for the gray fox. It was common in the early days, but the last few years we only got a few pictures. Coyotes are known to harass and kill their smaller canid cousin, and this seems to be resulting in a change in the Schenck Forest mammal community. Now, a lot of other NC State classes use Schenck Forest each year, but they are unlikely to see coyotes or foxes as they're walking around the forest during the day. If we hadn't set cameras to get the standard biodiversity monitoring data, we would have never known about these changes. There is so much biodiversity under our noses that we take it for granted, but if we don't pay attention, it could dwindle away. Then we would lose all the ecosystem services these species provide, endangering our own health and well-being as well. Biodiversity surveys provide the measuring stick. They describe what species live at a given place at a given time. If species start declining or moving away, these bioinventories can tip us off so we can help conserve biodiversity on our rapidly changing planet.